Okay, let's unpack this. Imagine chemistry as this giant three-dimensional jigsaw puzzle.
Right.
These aren't just flat pieces, you know.
Mm-hmm.
They're intricate shapes that fit together perfectly to make everything work, from, say, how you taste something sweet to how our bodies process new medicine.
Absolutely.
So today, we're diving into the fascinating world of molecular shapes, drawing insights from the core ideas in chemistry, the molecular nature of matter and change.
A classic test.
Exactly. Our mission today is basically to give you the mental tools to picture these tiny invisible architectures, understand why their shape matters so much, and connect this fundamental science to real-world stuff, all without needing, diagrams.
Yeah, think of it as visualizing chemistry.
Right. It's like your shortcut to understanding a molecule's real identity and how it behaves.
And what's really cool, I think, is that while we often start by drawing molecules flat, on paper,
Yeah, 2D drawings.
their real power, their function, it really emerges from their three-dimensional forms. So this deep dive should show how those 2D blueprints are just, well, the first step to unlocking the full picture.
Totally. And that first blueprint, that essential 2D map, that's the Lewis structure, right?
One way.
It's how we first sketch out where atoms are connected, showing which electrons are shared in bonds, the lines, and which are just hanging out as lone pairs the dots. It's our starting point.
It is. And when we build these Lewis structures, we're guided by a pretty fundamental idea, the octet rule.
Ah, yes, the octet rule.
Most atoms, they sort of strive to get 8 electrons in their outer shell for stability. Hydrogen's the exception, happy with the duet rule.
Right.
But this rule helps us logically place all the electrons in our 2D sketch.
Okay, so if we're drawing one, practically speaking, what's the first thing we do?
Well, the first mental step is just figuring out where to put the atoms.
The arrangement.
Yeah, the simple rule of thumb. The least electronegative atom usually goes in the middle. It's like the anchor. And remember, hydrogen, with its single bond capacity, it's never central. Once you've placed that central atom, you quickly add up the total valence electrons from all the atoms involved.
That's your electron count, your budget.
Exactly, your electron budget. You can't spend more than you have.
Makes sense. So we have the layout, we have the budget, then we start connecting things.
Precisely. You draw single bonds first, connecting the outer atoms to the central one. And for each bond you draw, you subtract 2 electrons from your budget.
Right.
Then you take the leftover electrons and distribute them as lone pairs. First, you give octets to the surrounding atoms.
Right, satisfy the neighbors first.
Yeah. And if there are any electrons left after that, they go on to the central atom. It's a systematic process.
But what if the central atom still doesn't have its octet after we've done all that?
Good question. That's the last key step. If that central atom is short, you need to form multiple bonds.
Double or triple bonds.
Exactly. You take a lone pair from one of the surrounding atoms and convert it into another bond, a double bond, or maybe even a triple bond if needed. Think of O2, oxygen gas.
Needs a double bond.
Right. Or N2, nitrogen gas, that famously has a triple bond. It's all about getting to that stable octet, usually.
That makes a lot of sense.
Yeah.
And if a molecule is bigger, like multiple central atoms.
Yeah, like ethanol or something.
Right. Do we just apply these same rules around each center?
Pretty much. You just handle each central atom sequentially, making sure the connections and local octets work out. You build it piece by piece.
Okay, got it. Now, here's where it gets really interesting, I think. What happens when, like, a single Lewis structure just isn't enough? It doesn't quite capture the reality.
Yeah, that leads us straight into
resonance. Resonance, where one picture isn't the whole story.
Exactly. Take ozone, O3. You can draw 2 perfectly good Lewis structures for it, and one the double bond is on the left and the other it's on the right. But here's the catch. Experimentally, both oxygen bonds in ozone are identical. They're the same length, same strength, something in between a single and a double bond.
Right. This is where that purple mule analogy comes in handy.
Yeah, the purple mule.
It's like a mule is a mix of a horse and a donkey, right? It's not a horse one second and a donkey the next.
It's a hybrid.
Exactly.
Yeah.
The actual ozone molecule isn't flipping back and forth. It's a resonance hybrid.
Yeah.
It's an average of those structures where the electrons are sort of smeared out, delocalized over the atoms.
Perfectly put, delocalized electrons. And because it's an average, the bonds aren't purely single or double. They end up with these fractional bond orders.
Like 1.5 for ozone.
Yep. Or 1.33 for the carbonate ion, CO32, where it's spread over 3 bonds.
So Okay, if we can draw multiple resonance structures, how do we figure out which one is, like, the best description or contributes most to that real hybrid?
Good point. That's where we use formal charge.
Calling charge, okay.
It's basically a bookkeeping tool. It's the charge an atom would have if all the bonding electrons were shared perfectly equally.
How do we calculate that?
There's a formula, but the idea is you compare the valence electrons an atom should have versus how many it owns in the Lewis structure, counting all lone pair electrons and half the bonding electrons.
Okay, and then we use that to pick the best structure.
Well, to pick the most important contributor, there are 3 guidelines, basically. 1, smaller formal charges are better, ideally 0.
Minimize charge.
2, avoid having the same non-zero charge on adjacent atoms, like 2 positives next to each other is bad.
Okay.
And 3, if you have to have a negative formal charge, it should be on the most electronegative atom. That atom is better at handling negative charge.
Makes sense. So these rules help us weigh the different resonance pictures.
Exactly. They guide us towards the structure that likely represents the most stable arrangement of electrons.
And it's probably worth clarifying, formal charge isn't the same thing as oxidation number, right?
Mm-hmm. Oh, definitely not. Good distinction. Formal charge assumes perfect equal sharing of bond electrons.
Right.
Oxidation number assumes the more electronegative atom takes all the bonding electrons. They're different ways of counting electrons for different purposes.
Gotcha. Okay. But chemistry, it loves to keep us on our toes. The octet rule is great, but there are exceptions, right? Especially like further down the periodic table.
Absolutely true. The octet rule is more of a guideline, especially beyond period 2. We see generally 3 main kinds of exceptions.
Okay, what's the first type?
First, you have molecules with electron-deficient atoms, usually central atoms like beryllium or boron, think Bf3. Boron only has 6 electrons around it.
Only 6, but it's stable.
It's stable enough to exist, but it's very reactive. It wants more electrons, so it readily forms bonds with other molecules that can donate a pair.
Okay, electron deficient. What's next?
Second exception, odd electron species. We often call these free radicals.
Free radicals. I've heard of those.
Yeah, they have an unpaired electron. Think of nitrogen dioxide, NO2. Nitrogen's in group 15, so it often ends up with an odd number. These are typically very reactive.
And this matters to us, right? Why are free radicals important outside the lab?
Oh, hugely important. They're linked to processes like aging, cell damage, even some cancers, antioxidants like vitamin E in our diet.
Yeah.
Their job is basically to neutralize these free radicals to stop them from causing damage. So yeah, very relevant biology.
Wow. Okay, so deficient atoms, odd electrons.
Mm-hmm.
What's the third exception? Going the other way.
Exactly. Expanded valence shells.
More than 8 electrons.
Yes. This happens with central atoms from period 3 or higher. Think phosphorus, sulfur, chlorine, xenon. These atoms are larger.
Bigger atoms, more space.
More space, yeah. And crucially, they have accessible d orbitals that can accommodate extra electron pairs. So you get compounds like PCl5, phosphorus pentachloride.
5 bonds. 10 electrons.
10 electrons around phosphorus, or
Yeah.
SF6, sulfur hexafluoride, 12 electrons around sulfur.
12. Okay, so the octet rule really breaks down there.
It does. Those d orbitals make all the difference for heavier elements.
So, okay, Lewis structures, resonance, exceptions. That gives us the 2D picture accounting for electrons. But you said earlier, the real action is in 3D. How do we make that jump from the flat Lewis structure to the actual, like, shape of the molecule?
Right. That's where VSEPR theory comes into play. VSEPR.
VSEPR, Valence-Shell Electron-Pair Repulsion. Sounds complicated.
The name is a mouthful, but the core idea is actually super simple. It just says that electron groups around a central atom.
Electron groups.
Yeah.
What counts as a group again?
Good clarification. An electron group is any region of electron density. So a single bond counts as one group. A double bond also counts as one group. A triple bond, still one group. And a lone pair, that's also one group.
Okay, so single, double, triple, lone pair, they each count as one zone of electrons.
Exactly, one zone. And VSEPR says these zones, these groups, will push each other away to get as far apart as possible. They want to minimize repulsion. It's all about maximizing space.
I like that balloon analogy you mentioned. Like tying balloons together, they just naturally spread out.
That's a perfect visual. They arrange themselves automatically into the lowest energy, most spread out configuration.
Okay. Now, you mentioned a key distinction. Electron group arrangement versus molecular shape. What's the difference there?
Ah, crucial point. The arrangement is determined by all the electron groups' bonding pairs and lone pairs. It tells you the overall geometry of those electron zones.
Like linear, trigonal, planar, tetrahedral.
Exactly. But the molecular shape only considers the positions of the atoms, the nuclei. It's the shape formed by the atoms themselves, ignoring the lone pairs when describing the final shape.
So the lone pairs influence the shape by pushing things around, but we don't see them in the final molecular shape name.
Precisely. They exert their repulsive force affecting the angles, but the shape is defined by where the atoms end up. We use the AXMN notation, A for central, X for bonded atoms, E for lone pairs to keep track.
And those ideal angles, like 180 for linear, 120 for trigonal planar, 109.5 for tetrahedral,
Yeah.
they don't always hold perfectly right.
No, they often deviate. And the main culprits are lone pairs and multiple bonds.
Why is that?
Well, think about a lone pair. It's only held by one nucleus, so its electron cloud is more spread out, more diffuse,
Takes up more space.
takes up more space, and exerts a stronger repulsive force than a bonding pair, which is stretched between two nuclei. So lone pairs tend to squeeze the angles between the bonding pairs.
Okay, so lone pairs push harder. What about double bonds?
Same idea, really. A double bond has more electron density than a single bond, so it also exerts a bit more repulsion. The order of repulsion strength generally goes lone pair is strongest, then lone pair, then bonding pair is weakest.
Got it. That subtle pushing and pulling fine-tunes the final geometry.
Absolutely. Let's maybe walk through the common shapes systematically.
Yeah, let's do it. Start with 2 electron groups.
Okay, 2 electron groups. They get as far apart as possible, which is 180 degrees. That's a linear arrangement. If both groups are bonds, AX2, the molecular shape is also linear. Think CO2, the double bonds count as one group each.
Simple enough. What about 3?
3 electron groups. They spread out in a flat triangle, 120 degrees apart. That's a trigonal planar arrangement. If all 3 are bonding groups, AX3, like in BF3, the shape is trigonal planar.
What if one is a lone pair?
If it's AX2E, 2 bonds, 1 lone pair, the arrangement is still trigonal planar, but the shape described by the atoms is bent or V-shaped. That lone pair pushes the 2 bonds closer than 120 degrees. SnCl2 is an example.
Okay, makes sense. Now for 4 groups. That's where 3D really kicks in.
It does. 4 electron groups arrange themselves in a tetrahedral arrangement, pointing to the corners of a tetrahedron. The ideal angle is about 109.5 degrees.
The classic methane shape.
Exactly. CH4 is AX4, pure tetrahedral shape, but replace one bond with a lone pair, AX3E, like ammonia, NH3, and that lone pair pushes the 3 hydrogens down into a trigonal pyramidal shape. The NH bonds are closer than a 109.5.
And if you have 2 lone pairs, like water.
Water, H2O, is AX2E2. Still a tetrahedral arrangement of electron groups, but those 2 lone pairs push the 2 hydrogens even closer together. The resulting molecular shape is bent or V-shaped with an angle even smaller than in ammonia, around 104.5 degrees.
You can really see how those invisible lone pairs dictate the final atomic structure.
They really do. Now, for 5 and 6 groups, we need central atoms from period 3 or higher.
Right, the ones that can have expanded octet.
Exactly. 5 electron groups give a trigonal bipyramidal arrangement. This one's a bit tricky. It has 2 different positions. 3 spots around the middle, the equator, at 120 degrees.
Equatorial.
And 2 spots directly above and below the poles, called axial positions, at 90 degrees to the equator.
Okay, axial and equatorial. Does it matter where lone pairs go?
It does. Lone pairs prefer the equatorial positions because there's more space there, fewer 90-degree repulsions. So AX5, like PCl5, is trigonal bipyramidal. AX4E, like SF4, puts the lone pair equatorial, giving a seesaw shape. AX3E2, like BRF3, puts 2 lone pairs equatorial, resulting in a T-shaped molecule. And AX2E3, like the triiodide ion I3, has 3 lone pairs equatorial, leaving the atoms in a linear shape.
Wow, quite a variety from just 5 groups. What about 6?
6 electron groups is more symmetrical. They arrange octahedrally, pointing to the corners of an octahedron. All angles are 90 degrees.
Like SF6.
Precisely. SF6 is AX6, a perfect octahedral shape. Now, replace one bond with a lone pair, AX5E, like in IF5. The lone pair can go anywhere, really, and you get a square pyramidal shape, a square base of atoms with one atom on top.
Okay.
Replace 2 bonds with lone pairs, AX4E2, like in X4. To minimize repulsion, the 2 lone pairs go on opposite sides of the central atom.
As far apart as possible.
Exactly. Leaving the 4 atoms in a flat square, that's a square planar shape.
That's a fantastic tour of the shapes. And just quickly, For bigger molecules with multiple central atoms, like, say, ethanol.
Yeah, you just figure out the shape around each central atom using VSEPR, and then you kind of mentally piece them together. So for ethanol, CH3CH2OH, the first carbon is tetrahedral, the second carbon is tetrahedral, and the oxygen is bent. You combine those local geometries.
It's like linking building blocks. Okay, so we have the shapes. What does this mean for how molecules behave? You mentioned polarity earlier.
Yes. This is where it all connects. Molecular shape is absolutely critical for determining if a molecule is polar or nonpolar overall. And that polarity dictates so much boiling points, solubility, how drugs interact in the body.
It's not just about whether the individual bonds are polar.
Exactly. You can have polar bonds where electrons aren't shared equally because of electronegativity differences, but still end up with a nonpolar molecule.
How did that happen?
It's all about symmetry. Take CO2 again. Each CO bond is polar. Oxygen pulls electrons more than carbon. But CO2 is linear. The two polar bonds point in exactly opposite directions. So their poles cancel each other out, like a tug of war where both sides pull equally hard. The net result, zero overall polarity. CO2 is nonpolar.
Okay, cancellation due to shape. What about water?
Water, H2O. The OH bonds are very polar, but water has that bent V shape because of the lone pairs.
Right, not linear.
Because it's bent, the poles from the two polar OH bonds don't cancel. They add up, creating a net direction of pull, a molecular dipole moment. Water is very polar.
So shape prevents cancellation. That makes sense.
Another great example is comparing cis and trans 1-methyl-2-dichloroethylene. Same atoms, same bonds.
Just arranged differently around the double bond.
Exactly. In the trans isomer, the two polar CCl bonds are on opposite sides, pointing away from each other. They cancel out. Trans is nonpolar.
Okay.
But in the cis isomer, the two CCl bonds are on the same side. Their poles add together. Cis is polar.
Same formula, different shape, different polarity.
Precisely. And this has real consequences. The polar cis molecules attract each other more strongly than the nonpolar trans molecules.
So the boiling point is different.
Significantly. The cis isomer boils at about 60 degrees C, while the trans boils lower, around 48 degrees C, all because of the shape influencing polarity.
That's a clear connection. And, thinking bigger picture, this importance of shape, it's absolutely massive in biology, isn't it?
Oh, profoundly important. You could say molecular shape is the basis of biological recognition. Living systems are all about molecules fitting together correctly, like a lock and key.
That lock and key idea, that applies to our sense of smell, right?
Perfectly. For you to smell something, an odor molecule has to physically fit into a specific receptor protein on the nerve endings in your nose.
It has to have the right shape.
The right shape, or at least a part of it has the right shape. Axel and Buck won the Nobel Prize for figuring out we have maybe 1,000 different types of these receptors.
Yeah.
And by combining signals from different receptors activated by different parts of a molecule or by similarly shaped molecules, we can distinguish something like 10,000 different smells.
So different molecules might smell the same if they have a similar shape that fits the same receptor.
Exactly. That's why different chemicals can all smell, say, camphor-like. They share a shape feature recognized by the camphor receptor.
Fascinating. And this lock and key thing, it's not just smell, it's everywhere in biology.
Everywhere. Enzyme action. The substrate molecule has to fit the enzyme's active site. Nerve signals. Neurotransmitters fit into receptors on the next nerve cell.
Like puzzle pieces.
Immune system. Antibodies recognize pathogens by the shape of molecules on their surface. Hormones. They only work when they fit into the receptors on their target cells. Even DNA function relies on proteins recognizing specific shapes along the double helix. It's shape, shape, shape.
It really is the architecture of life at the smallest scale. So, wow. Today we've gone from those flat Lewis diagrams
The starting point.
to understanding how electron pairs push each other around with VSEPR theory, creating these amazing 3D shapes. And we saw how that shape is key to polarity, which affects physical properties, and ultimately how biological systems function through molecular recognition.
From simple repulsion rules to the complexity of life.
Key takeaways seem to be Lewis structures give the connections, VSEPR gives the 3D geometry based on minimizing repulsion, and that geometry determines polarity and function.
You've nailed it. Arrangement, shape, polarity, function, it's a direct chain.
So reflecting on all this makes you wonder, doesn't it? As we get better at understanding and even predicting these molecular shapes, what does that unlock for the future?
That's a great final thought. I mean, it raises the question, right? If we can truly master predicting and designing shapes, What does that mean for creating targeted medicines with fewer side effects? Or designing new materials with specific properties? Or maybe even understanding at a deeper level how life itself might have originated from molecules recognizing each other?
Huge possibilities. Well, that seems like a perfect place to wrap up. A massive thank you from the Deep Dive team for joining us on this exploration of molecular shapes.
My pleasure.
Keep exploring, keep questioning, and stay curious.